Hello everyone. Today we are going to discuss about the topic bioremediation, concept, need, scope and applications. Myself Arshida A, first MSc Biology, St. Mary's College, Thrissur. What is bioremediation? Remediate means to solve a problem and bioremediate means to use biological organisms to solve an environmental problem such as contaminated soil or groundwater. To use a biological remedy to abate or clean up contamination. Basically, it is a waste management technique that involves microorganisms to remove or neutralize pollutants from contaminated sites. Bioremediation is a biological degrading process for the treatment of contaminated soils, groundwater, sediments, relying on microorganisms, including bacteria or fungi, to use the contaminant as a food source with resulting degradation of the contaminant. Microorganisms used to perform the function of bioremediation are known as bioremediators. Most economic remedial techniques presently available for treating most organic fuel-based contaminants such as coal tars and liquids, petroleum and other carcinogenic hydrocarbons such as naphthalene, benzene and some inorganics. The basis of removal and transportation of waste for treatment. Basically, there are two methods: in situ bioremediation and ex situ bioremediation. In situ means at the site. The treatment of contaminated material in a place. Example, benzene, toluene, TNT 24D. Here, complete degradation is often difficult to achieve. Ex situ bioremediation takes place away from the site, and these techniques involve physical removal of the contaminated material for treatment process. Example, biopiles, soil treatment unit, compost pipe, etc. And this technique involves the use of bioreactors to process the material in a highly controlled environment. How does it work? First, waste material is examined. Certain bacteria are isolated based on their efficacy in digesting and converting the waste. Indigenous or local bacteria is to be used. And then bacteria then go through several steps of cultures and process for performance testing. Suitable bacteria are placed back in the waste environment. They grow and thrive in the process, digest and convert the waste into carbon dioxide and water. Right temperature, nutrients and food also must be present. Conditions may be improved by adding amendments. What are the essential factors for microbial bioremediation? First one, microbial population, and the desired condition is suitable kinds of organisms that can biodegrade all of the contaminants. Next factor is oxygen, enough to support aerobic biodegradation, about 2% oxygen in the gas phase or 0.4 mg per liter in the soil water. Next, water. Soil moisture should be from 50 to 70% of the water holding capacity of the soil. Next factor, nutrients. Nutrients such as nitrogen, phosphorus, sulfur and other nutrients to support good microbial growth. Temperature. Appropriate temperature for microbial growth is 0 to 40 degrees Celsius. pH. pH is from 6.5 to 7.5. Bioremediation of soils. Bioremediation of soil involves two processes. First one, biostimulation. Basically involves the stimulation of microorganisms already present in the soil, and this can be done by various means. Addition of nutrients such as nitrogen and phosphorus that promote microbial growth. Supplementation with the co-substrates. Example, methane added to degrade trichloroethylene. And addition of surfactants to disperse the hydrophobic compounds in water. That is, expose hydrophobic molecules from the soil. The next process is bioaugmentation, and this involves the addition of specific microorganisms to the polluted soil constitute bioaugmentation. Here, pollutants are very complex molecules, so native soil microorganisms alone may not be capable of degrading them effectively. Pollutants such as polychlorobiphenyls, trinitrotoluene, polyaromatic hydrocarbons and certain pesticides. Based on research findings at laboratory level, now possible to add a combination of microorganisms that is referred to as consortium or cocktail of microorganisms to achieve bio-augmentation. With the development of genetically engineered microorganisms, they can also be used to bio-augment soils for every efficient bioremediation. But the direct use of genetically engineered microorganisms in the soil is associated with several risks and health hazards. Techniques of soil remediation. Most commonly used methods for the bioremediation are in situ bioremediation, land farming, slurry phase bioreactors. In situ bioremediation broadly involves the biological cleanup of soils without excavation. This technique is used for the bioremediation of subsurfaces of soils, buildings, and the roadways that are polluted. Sometimes water is cycled through the subsurfaces for increasing the efficiency of microbial degradation. There are two types of in situ soil bioremediation techniques. First one, bioventing, very efficient and cost-effective technique for the bioremediation of petroleum contaminated soils. Involves aerobic biodegradation of pollutants by circulating air through subsurfaces of soil. It takes some years. Bioventing can be used for the degradation of soluble paraffins and polyaromatic hydrocarbons. And major limitation of this technique is air circulation, which is not always practicable. Phytoremediation. This technique involves by the use of plants. Specific plants are cultivated at the sites of polluted soil. These plants are capable of stimulating the biodegradation of pollutants in the soil adjacent to roots. Phytoremediation is a cheap, environment-friendly cleanup process for the biodegradation of soil pollutants. It takes several years. Land farming in soil remediation. And this is a technique for the bioremediation of hydrocarbon contaminated soils. Soil is excavated, mixed with the microorganisms and nutrients and spread out on liner just below the polluted soil. Soil has to be regularly ploughed for good mixing and aeration. If the soil is mixed with compost and or temperature is increased, the efficiency of biodegradation increases. Addition of co-substrates and anaerobic pre-treatment of polluted soils also increases the degradation process. Land farming has been successfully used for the bioremediation of soils polluted with chloroethane, benzene, toluene, and xylene. These three compounds are always known as BTX aromatics. Slurry phase bioreactors in soil bioremediation. And this is an improved land farming system. Excavated polluted soil is subjected to bioremediation under optimally controlled conditions in specifically designed bioreactors. Due to close contact between the xenobiotics and the microorganisms and the optimal conditions, degradation is very rapid and efficient. Slurry phase bioreactors are, however, not suitable for widespread use due to high cost. Bioremediation of groundwater. Two techniques are involved. First one, pump and treat technique. And this technique is based on physico-chemical principles to remove pollutants. Treatment units are set up above the ground. Strip columns and activated carbon filters can remove most of the groundwater pollutants. Treated water is recycled through injection well several times so that the pollutants are effectively removed. For removal of certain organic pollutants, bioreactors have to be installed. Biodegradation of a tetrachloroethane, a bioreactor with granular methanogenic sludge, is found to be effective. In recent years, bioreactors with both aerobic and anaerobic bacteria have been developed for better bioremediation of highly polluted groundwaters. However, not possible to achieve good cleanup of groundwater by pump and treat technology because of subsurface heterogeneity, strongly adsorbed compounds, and low permeability of pollutants. Biofencing. This is an improved technique. Consists of installation of a bioactive zone at the down gradient edge of contaminated groundwater area. Nutrients are injected through a well to the bioactive zone. As the groundwater passes through the bioactive zone, the pollutants are biodegraded and clean groundwater comes out. Applications used for the remediation of metals, radionuclides, pesticides, explosive fuels, volatile organic compounds, and semi-volatile organic compounds. And research is underway to understand the role of phytoremediation to remediate perchlorate, a contaminant that has been shown to be persistent in surface and groundwater system. It may be used to clean up contaminants found in soil and groundwater. For radioactive substances, chelating agents are sometimes used to make the contaminants amenable to plant uptake. Thank you.